Introduction/neuroanatomy. Hi, my name is Angelica, and Luke Norena is the coolest person ever. The human neuron is a cell found in the nervous system responsible for the transmission of electrical and chemical information throughout the body's nervous system. The dendrites are short branched extensions of the neuron along which information is received from other cells within the body. These extensions are crucial for the reception of information throughout the human body. Coming off of the dendrites are small membranous projections called the dendritic spines.
Wow!
These are tiny specific extensions of the dendrites that receive electrical and chemical information from the presynaptic neuron. Both the dendritic spines and the dendrites are attached to the cell body of the neuron. The cell body is also called the soma. The soma is a round-shaped part of the neuron that contains the nucleus. It acts as the factory of the neuron, helping all of its parts perform their function. Directly adjacent to the cell body is the axon hillock. This part is the final extension of the soma that connects the cell body to the neuron's axon. The axon hillock is crucial for the transfer of electrical inputs from the dendrites and the cell body to the axon. This is where an action potential is initiated. From the axon hillock, the action potential then gets transferred down the axon to the axon terminals where the neurotransmitters are released, producing the transmission of information. Neurotransmitters have signals that are either excitatory or inhibitory. Excitatory signals depolarize the cell, while inhibitory signals hyperpolarize the cell. The resting membrane potential of the cell is approximately -70 millivolts, and once there is enough excitatory signals inside the cell, sodium channels open, allowing sodium to flow into the cell and begin the depolarizing phase. Once it has reached the threshold at -55 millivolts, the action potential is sent. The depolarizing phase continues until 30 millivolts, which is when the sodium channels close and the repolarization phase begins. In the repolarization phase, the potassium channels are open and potassium is flowing out of the cell. So much potassium flows out of the cell that there is a brief period of hyperpolarization, and this is when the sodium-potassium pump kicks in. Sodium-potassium pump is an active transport that is ATP-driven. 3 sodium ions bind to the transmembrane protein, and the protein becomes phosphorylated at the expense of an ATP molecule. Phosphorylation induces a conformational change, and 3 bound sodium ions dissociate from the protein and are now located on the extracellular side. This open conformation now has a high affinity for potassium ions, and 2 potassium ions bound to the transmembrane protein. The bound phosphate will dissociate, and the transmembrane protein changes conformation once again, and the 2 bound potassium ions dissociate from the protein into the intracellular portion of the cell.
Another way in which an action potential can be manifested is through spatial summation. When multiple presynaptic neurons produce either EPSPs or IPSPs, these forces can be summed together over space to initiate an action potential. The depolarizing forces in a space sum together to generate a force strong enough to exceed the threshold of the postsynaptic neuron and therefore initiate an action potential.
Temporal summation occurs when the presynaptic neuron releases neurotransmitters multiple times over a period of time. Over time, the effects of neurotransmitters being released from the presynaptic neuron sum together to cause a depolarizing force that exceeds the threshold value of the postsynaptic neuron. The graded potential forces can temporarily sum together to manifest an action potential.